Swayam Prabha. Digital India. Educated India.
Today's lecture we will discuss about the construction of phylogenetic trees. Right? Yesterday's class, so we discussed various aspects on conservation. So could you please remember something what we discussed in the last lecture?
The frequency.
Yeah, right. So what is the meaning of conservation?
So how far a residue is conserved in a
Right, in any protein sequence, how far a specific residue maintains at the same position in different organisms, in different homologous sequences, whether it maintains the same residue at the same position or it changes with respect to different organisms or with respect to different time. So, if it is same, then we call that this residue is conserved at the particular position. So, how to understand or how to derive, how to qualitatively measure the conservation of the residues at different positions in a sequence. In this case, it is a two-step procedure. What is the first step? Yeah, we try to calculate the frequency of occurrence of amino acid residues at any particular position, right. Then we convert these frequencies into scores. For getting the frequency of occurrence, there are different ways to get the frequency of occurrence. What are the different ways to get the frequency of occurrence? Unweighted frequencies. In this case, we do not give any weightage to any sequence or no weightage to any amino acids. All residues and all sequences are treated equally. So, it is useful if you compare a closely related sequences. In this case, most of them are from homologous families and several sequence residues are the same. So, it is useful for the closely related sequences. Then what is another method to calculate the frequency?
unweighted.
Weighted frequencies, right. What to do with the weighted frequencies? No, we give different weightage, any specific weightage for any specific residue which like to be maintain the same position, then we give weightage, or any particular sequences. For example, if we have many sequences from closely related ones, homologous sequences, and some of them from distantly related ones, then if you give weightage, right, to include the information from distantly related ones, then we give weightage. So, the third one we discussed about the independent counts. So, how randomly distributed with respect to your original distribution, right, you can get independent counts to get the frequency. Then when the frequency is calculated, then we convert these frequencies into scores. So, we discussed about different types of methods to get scores. What are the different methods to calculate the score? Entropy based, variance based measure, sum of pairs method, right. In the case of the entropy based method, how to work? So, you get the probability of the particular amino acid residue, that is frequency of residues at any particular position, multiplied with this logarithmic amount. So, it will give you the entropy based method. In the case of variance based method, it gets information for the same amino acid residues at different positions, how far they change, how far they maintain at different positions in an amino acid sequence. So, you get the information. In the case of sum of pairs method, what we do? We take these pairs and then we can use any matrix. Okay, so, you can normalize any matrix and you can use any matrix to give weightage to similar residues compared with the residues of different kinds. So, we get the conservation. Then we discuss about the normalization. So, we can use different normalization procedures to normalize the scores. In the case of entropy based method, if a particular position is occupied with the same residue, what is the score? 0, right, because the frequency is 1. So, 1 in logarithmic of 1, that is equal to 0. So, you get the numbers, negative values for the variable ones. So, you can also normalize, right, based on 0 to 1 or based on this average value of 1 and so on. So, when we discussed about few online resources, what are the online resources we discussed to calculate the conservation score? AL2CO, right, and then conserve. What is the input for the AL2CO server? Alignment. You give the multiple sequence alignment. So, it takes multiple sequence alignment and you give the score. You give all the options, you can choose any of the options to get the score. Then for the conserve, You can give the PDB ID, right? It will automatically get the sequences and do the alignment and get the consensus score. So, today if you have these sequences, right, we discuss about a multiple sequence alignment. There are many sequences and which sequences are close to each other and how far it takes to change one sequence to other sequence with respect to different organisms. So, in this case we can draw a tree type of method, right, make a trees to show which residues are similar to each other. So, phylogeny if you define, it is a description of the relationship, any biological relationship, right, expressed as a in the form of tree. For example, if you take the human genealogy, say the grandfather here, grandmother, so they have several children. So, now if you see he is the father, likewise you get from mother tree you can get the mother. So, here they have several children, right. So, you have this is maybe you, this is your siblings, your brother, sister, so on. Here also you can have different things. So, like this for the different sons or daughters of these grandparents. Then here you are here. So, you have your son, your daughter and so on, right. So, you have these relationships. To understand the relationship, for example, whether you can see any similarities in the shape or any of these similar organs, right, if it look similar, then you can see they may be close to each other. From this to this you can easily know that, and from it here you also have a relationship, and here to grandmother daughter, my daughter to the grandmother, some possibilities. Then if you go for the more and more generations, then we do not know. So, but you can listen that okay, he looks like his grandfather, right, he looks like his aunt. So, how to do this? There are two different ways. One is look like the shape or you look like any of the organs, or then depends on time. How long it took from one generation to other generation. So, you can understand two different aspects. One is you can see the time from one generation to another generation, and also you can see the relationship, how far the two people are related to each other, on what aspects. So, if you took the close related ones, you can see much similarities. When it goes further and further, you can see similarities, but less compared with the close related ones. Likewise, you can see the phylogeny. So, this is among different objects, which assumes the homology because they are similar to each other, and this also depends upon how they classify. So, then this analysis, what is the phylogenetic analysis? It is investigation of this evolution relationship, right, from one organism, different organisms among the group of related sequences by giving a tree like of representation, just we discussed earlier. So, we give a tree like of representation to discuss the relationship, to understand the evolution relationship. In fact, there are various ways to explain the relationship, but this is the easiest way, right. For example, if you say A and B are related to each other, B and C are related to each other, A and D are related to each other. So, you can explain different ways, but the tree wise representation is the easiest possible way to understand the relationship among different organisms. So, if we see the organisms with high similarity, like it just I discussed earlier, they are closely related. So, then they are dissimilar. So, in this case the ones which are closely related, they put nearby each other. So, you can easily understand okay, these two are close to each other. So, due to the availability of data, the taxonomists first they started to construct trees, right. So, they rely on the comparison of phenotypes, like how the organisms look like, and to infer the genotypes, right, what gives this type of appearance. Likewise, in the family tree as I showed earlier, so you can see look at this how they look like, then they will try to compare okay they look like, so these two are similar to each other, right. Then they try to do this, but when it happens to the various systems, for example, if they take the light detecting organ eye to see the common behavior of these different organisms, then sometimes it fails because this is eye we can see from humans, flies, mollusks and so on. So, then they try to use different types of information to infer the sequences which are similar to each other, the organisms which are similar to each other. So, currently due to the availability of different sequences, or protein sequence or DNA sequences, it is easy and it is reliable to compare the sequences and identify the lineage between different organisms, because we have the different sequences for DNA, right. Where you can get the DNA sequences? GenBank or EMBL or DDBJ and so on. So, as you get the protein sequences, UniProt is a unique resource, right. We get the protein sequences. So, currently also we get different sequences from several organisms, right, and the sequences are also reliable. So, easily you can compare these sequences to understand the evolution relationship and how far it took from one organism to different organism, and how about the different variations in the sequences and so on. So, now if we have the sequences, we can construct trees to show the evolution relationship of any genes or organism, at least 3 or more genes or any organisms. For example, if you have 3 organisms A, B, C, if A and B are similar to each other with minor change, for example, if there is a 2 amino acid change, right, and B and C are also similar to each other, which are 5 amino acid difference, and you can see A and C are similar to each other, for example, you can see the 10 amino acid difference. Then if you have 3 cases, one is A, one is B, one is C, which are close to each other, E and B are close to each other because we have the mismatch of only 2 residues, right. Likewise, if you have more number of data, 3 or more sequences, from the differences in the nucleotides or in the amino acids, you can see how they are related, which 2 are close to each other, and how long it takes to get this divergence. Depending upon the variations, we can estimate the time. Okay, this much time, right, takes to get this convergence, to get the divergence times from one organism to other organisms. Then you can see the nature of common ancestor, right. If you make it tree, you can see what will be the common ancestor. For example, here A and B are close to each other, so they may have a common ancestor, right. Likewise, you can see the common ancestor when you construct phylogenetic trees. So, how to represent trees, how to draw trees. So, I show one example here. So, if you have a common ancestor here, this is the one, D is here. So, then from here I take the trees. So, this goes from number 5 through C, we can see 3 and 4, right, and through A you can see 1 and 2. When you make a tree, for example, this is A, this is B, both are common to each other, okay, you can connect these two. So, if you see this figure, so which one are close to each other?
1 and 2 and 3.
1 and 2 are close to each other, and 3 and 4 are close to each other, right, and this 1 and 2 and then 3 and 4, it takes time, but they have some similarity which is a common with C, and then all these things 5s, so they will be connected together at the point D. So, when you draw trees, there are 2 different aspects. One, we can see the dots and you can see the lines. So, lines represent branches and the dots represent nodes. You can see this node and this line, they are branches. And if you see the nodes, there are different types of dots here. You can see 2 different types of circles. One is a closed one here, 2, 3, 4 and 5, and also you can see some of the nodes which are in the interior, there is open circles here, okay, this and this. So, here these 2 circles, they represent 2 different aspects. The one which at the end, tip of the branches, they are called the terminal nodes, okay, these are called this is called terminal nodes. Like you can see the gene or any organism or any sequence, that's we know the data, that information is available, and you can see the another dots inside ones, and here these are the internal nodes, okay, for example, these are internal nodes which represent common ancestor, but the information we do not know, because if you look into the UniProt database or if you look at DDBJ database, you will get the sequences, that information we know. Then which 2 are common and which 2 are emerged in specific period of time, that we do not know. So, when you make this type of trees, you can try to understand which organisms are close to each other and which organism emerges first and how long it takes to have the next one and so on. So, if you have this type of trees, there are 2 types of trees. For example, if you see this node, it has 2 branches, some cases you have 3, some cases you can 4. So, if there is 2, then this is called the bifurcating ones, the nodes. So, there can be more branches. So, this one we called as multifurcating. So, right here we have 2, for example, if you have another one here, that will be 3 or 4. So, in this case you can distinguish the bifurcating as well as the multifurcating. And another aspect is, in some cases you know how long it takes from one organism to another organism, some cases we do not know. So, this will tell you the scaled trees and the unscaled trees. If you take 100 years or 10,000 years, so if you can scale it, then this is called scale trees, and if you do not know the time, then that case you can call this as unscaled trees. So, now if you have a trees, right, making long trees, right, that like space. In this case you can simplify the trees in the form of specific notations, like you can use some parentheses. If you write the parenthesis, this will tell you how the tree looks like, and this format is called the Newick format. How to write this Newick format? For example, if you see this tree, 1 and 2 are close to each other. So, 1 and 2 are close to each other, and then 3 and 4 are close to each other, right, 3 and 4 they are close to each other, and these two, this is close to each other, this is this node A, and here these are close to each other, this node B, but these two are close to each other. So, now you can say these two are close to each other. Now, the next one is the number 5, 5 is alone. So, 5 is here, and this is, and this 5 are connected with each other. So, you can see another bracket here, then we can see this is the format. So, go to the internal brackets, then you can construct a tree. For example, 1 and 2 is connected, 3 and 4 connected, and these 2 are connected with each other, and 5 is connected with everyone. So, there is a format which you can write represent trees using these parentheses. Okay, this format is called the Newick format, right. If you have a trees, you have nodes, you have branches, 2 types of nodes, terminal nodes and internal nodes. Terminal nodes represent any genes or any organism, so data are available. Internal node provides information on common ancestor that we do not know. When we construct the trees, then we can have the information, and there are scaled trees and unscaled trees. What is the difference between scaled and unscaled trees? Time, we know the time or we do not know the time, right. Then we have the, depending upon these branches, we have bifurcating and multifurcating, right, and you can write these trees in a form of a specific format. This is called the Newick format. Now, there is another type of trees that is called rooted trees and the unrooted trees. What is rooted trees and what is called unrooted trees? The rooted trees means you can see a single node which is designated as a common ancestor. So, in this case we know the ancestor, but the lineage also we know, but we do not know the relationship, but we have the ancestor and we have the path to lead to this tree. All these 1, 2, 3, 4, 5 finally comes to this point in specific period of time. That is called rooted tree. So, rooted tree, a single node is designated as a common ancestor. In the case of unrooted trees, we know the relationship, but we do not know the direction. For example, 1 and 2 are related and 4 and 5 are related. So, all they are related, but where they starts, where they ends, we do not know, but they are related to each other, right. So, for example, if you see a group of people, you can say okay, they look similar, so maybe related to each other, but I do not know where we have the ancestor, maybe 4th generation, 5th generation, that we do not know. Some cases we know you look similar, but we know the ancestor, okay, your forefather or your grandparent was he. So, you both from the thus different linealogy, right. This is how we can see the rooted trees as well as the unrooted trees. Okay, now for example, if you have the 3 species, right, 1, 2, 3, how many times you can have the rooted tree and how many times you can have the unrooted trees, right. If you have 3 species 1, 2, 3, if the unrooted tree we do not know the common ancestor, right. In this case you can draw in any way, whatever you draw, so you will get only one tree, right, because we do not know about ancestor. But in the case of rooted ones, ancestor could be 1, ancestor could be 2, ancestor could be 3, right. In this case you will have 3 different ways to construct these rooted trees. So, this is 1, this is 2, so you have the third one, right. Here is 1, here 2 is here and 3 is here, and then it emerges 1 and 2. Here first one is here and second and third. here first is the second one, this is the common one and first and third. There are 3 different ways you can have the rooted trees, right, if you think about the ancestor. Likewise, if you have 2 data, how many rooted trees and how many unrooted trees? 1 rooted trees and 1 unrooted trees. If you have 3 data, 3 rooted trees and 1 unrooted tree. If you have 4 data, depending upon the first one, you will have 15 rooted trees and 3 unrooted trees. So, this is the equation used to calculate the number of rooted and unrooted trees depending upon the organism. So, n is the number of species, and you can use this equation 2n minus 3 factorial divided by 2 into n minus 2 in n minus 2 factorial. So, this is the equation we can fit these numbers here. So, number of rooted trees and the number of unrooted trees. For example, if you have 3 data, how many number of rooted trees? 6 minus 3, that is equal to 3 factorial, that is equal to 6 divided by 2, right. So, 3 minus 2 equal to 1, here equal to 1 into 1 1. So, this is equal to 3 by 1, that is equal to 1, right. So, here n equal to 3, then this case n are equal to 3, right. So, n equal to 3. So, number of rooted trees equal to 3, you can use this equation. Likewise, if you say unrooted trees, this is n equal to 3, then nu equal to 1, right, you can fit this equation. Likewise, you can use the 4 data or 5 data, you can get the number of rooted and unrooted trees. And this question I think once they asked in the GATE exam. So, how to relate number of rooted and unrooted trees.